In part 1, we've looked at inheritance, the gene, and cell division. Okay, part 2 part 2 we'll be looking at the gene and protein, genes and proteins. Now, first of all, let's take a look at the DNA-based sequence to amino acid sequence. Okay, this is for IGCSE students, those who are in science stream. Okay, so you know the combination of the base sequence of a DNA will form amino acid, the base sequence of an amino acid. So we say the DNA base sequence determines the amino acid sequence in protein. So combinations of the base sequence, the code, the DNA code of the base, combination of it forms the base of the amino acid. And super amino acid, many amino acid together, combination of amino acid or series of amino acid forms the protein. So we say the DNA code, which is the code itself. When we say a DNA code, we refer to the series of the base, the series of the bases of the DNA strand. We are referring to the series of base. So when we say DNA code, we refer to series of bases of the strand, of the DNA strand. Now this DNA code, which is a series of base, it's converted to protein. How? Protein is a series of amino acids. That's just what we call protein. Because this DNA code, which is the series of the base, combined together forms the series of amino acid. Combined together, many of the base, when you code them together, they will form the series of amino acid. And it is this series of amino acid that we call protein. Why do we call the series of amino acid? Because each got its own shape. So when they combine, they form a protein. So a protein is made up of a series of amino acid coming from the series of the bases of the DNA strand, or the series of bases of the DNA strand. Now, the process of protein synthesis has two stages. To form a protein, the basis or the DNA code, we have to undergo two processes. The DNA coding must go through two processes. Now, the first one is called transcription. Now, what do we mean by transcription in the formation of protein? You know, like I said, protein is life. Okay, protein is life, is responsible for the formation of enzymes. Proteins are enzymes. Proteins they form most of the hormones and most of the things in our body. So if you don't have enough protein to produce your system, or you don't have the coding, enough code to make amino acids and proteins in your body, then there are a lot of series of problems that will result, which we're going to talk about later on. Some of these could be like serious damaging effects. Like you see children born these days, a lot of them has this autism problems and all of this. So when there are not enough proteins to code all these parametric behavior, all these parameters in the metabolic activities in our body, then we start having deficiency and a lot of problems in the whole formation. So number one process of forming protein, or number one process of protein synthesis, is transcription. Now, what is transcription? Transcription is just a way of writing again. You're writing something again. You're rewriting the base code of a DNA into bases of RNA. We know what is DNA, right? Okay, so the process of rewriting the base code of a DNA into what we call the basis of RNA, ribonucleic acid, nucleic acid for RNA. So the process of rewriting the code, okay, a code that is already written for a DNA, you're rewriting it into basis of RNA. That process is called transcription. So please we keep in mind, rewriting the base code of a DNA into RNA is called transcription. You are transcribing. Okay, the process is called transcription. The second process of the synthesis of protein or of protein formation. Okay, protein manufacturing or protein formation or protein synthesis, whatever language you want to use. The second process is called translation. The second process is called what? Translation. Now, what is translation? Translation is now, you formed an RNA. When you transcribe the base code of a DNA into RNA, is called transcription. Now, translation is now you're using the RNA base sequence to build amino acid into sequence in a protein. You are now using the RNA. So it's before protein can be formed or can be synthesized. First of all, the DNA base code will be transcribed into RNA base sequence. Now, the RNA base sequence will be used to build amino acid, which will form a sequence of protein. Okay, so that second process, the first process is to change the DNA base sequence into RNA. That's transcription. Translation is now when you're using the RNA base sequence to build amino acid sequence that will eventually become the protein. That's what we call translation. So I hope we get these two things clear. So it's very important we understand the two processes that is undergone when protein is being formed, when protein is being synthesized. Okay, we need to get this very clear. Okay, transcription and translation. Transcription is rewriting the base code of a DNA into an RNA base sequence. Translation is now using that RNA base sequence to build amino acid, which will eventually become the sequence of our protein. Now, so the sequences of the base in a okay, if we want, we can show something, right? We can show something. We say transcription is simply transcription in genetics or in transcription in DNA study or protein formation. The synthesis of protein, number one, is transcription. We say this is when the base code of a DNA, the base code is rewritten. Base code of a DNA is rewritten. Base code of DNA is rewritten to form RNA base sequence. So base codes of a DNA, the base code of a DNA sequence, okay? The base code of a DNA sequence, the process of rewriting it to form RNA. You already have the code. The code of the DNA has already been written by the creator of human being. Now, the process whereby your body mechanism rewrite the base code of a DNA sequence, okay, when it's rewritten, okay, past participle form of rewrite, rewritten, double T-E-N, okay, is rewritten when the base code of a DNA base sequence is rewritten to form RNA, to form RNA base sequence. So this is called transcription. So we should know this in exam. This is called transcription, RNA base sequence. When it's ready to form RNA base sequence, this is called transcription. Okay, we need to keep this in mind. It's very easy. The base code, a DNA is actually from the base code, series of the base code. Okay, series of base code that will combine to form amino acid. Okay, and this series of amino acid is what we call the protein. So this base code or base sequence is the DNA. Is that okay? The base sequence is the DNA. Now, when the base sequence combine like this, the base here combine, the base sequence combine, it forms a DNA, and it forms a amino acid, series of amino acid, and this series of amino acid forms a protein. Now, before we get there, the first process is called transcription. Now, we say in transcription, it's when the base code of a DNA sequence is rewritten. Okay, you can put is rewritten to form the RNA base sequence. Now, the second process is called translation. The second process is called translation. Now, what is translation? Translation is simply the RNA, the process whereby the RNA base sequence. Translation is simply the process whereby the RNA base sequence join together to form the amino acid or the protein. Okay, the process whereby the RNA base sequence form the base sequence of the amino acid. Okay, so the process whereby the RNA base sequence, RNA, the N was notoriously big, beyond the intention. Okay, let's make the R also big to compensate for that. Okay, so the process whereby the RNA base sequence join to form the series of amino acid. Join to form the series of amino acid. Is that okay? So when the RNA sequence build the amino acid, like they join together to build the amino acid into a sequence in a protein. Okay, join to build, we can use the word join. Okay, join to build series or sequence of amino acid. To build sequence or series of amino acid that will form the protein. Sequence or series of amino acid, series of amino acid that will join together to form the protein. Okay, this is not H, this N. Amino acid, series of amino acid, to form a sequence of protein. To form a sequence of protein or series of protein. Okay, this is translation. Translation is now when RNA base sequence build what we call the sequence of protein. Okay, so that's translation. So these two processes are very vital for moving forward. Okay, now we say the sequence of bases in a gene determines the sequence of amino acid that makes a specific protein. The kind of sequence you have for the base determine what kind of amino acid, the base of amino acid that will be formed. For example, let's use shapes. If you have triangular shape base sequence of the amino acid of the base or of the code of the gene, it will form that triangular base of the amino acid, which will eventually be that shape of the protein for that specific protein. Because different amino acid has different shapes, Is that okay? Now that will form that specific protein, because we have protein for specific purposes. Now different sequences of amino acid gives different shapes and functions to protein molecules. See, different sequence of amino acid give different shapes and functions to protein molecules. So if you have different amino acid or sequence of amino acid, like for example, if the shape of the amino acid is rectangular, then the protein that will be formed will have a similitude. Okay, now let's go to the next protein synthesis, the process itself. So before we continue on the protein synthesis, let's listen to this tutorial before we now look at the protein itself, the DNA strand itself. Okay, before we look at the DNA strand itself, let's listen to this tutorial. Okay.
This video is on protein synthesis and is aimed at students studying the extended syllabus. Protein synthesis is all about making the proteins required by the body. This could include enzymes used to catalyze reactions, for example in digestion, or carrier proteins found in the cell membrane, which are used to transport particles across the membrane. Proteins are made within the cells in a two-step process. The instructions for building the proteins are all contained within the DNA code, which is found in the nucleus of the cell. However, it cannot leave the nucleus of the cell. Therefore, transcription is required to copy sections of DNA into small strands of messenger RNA, or mRNA, which can leave the nucleus. Step 2 is translation, which occurs in the cytoplasm. This stage involves the conversion of mRNA to an amino acid sequence which forms the protein. The role of transcription is to produce short, single-stranded sections of mRNA using DNA as a template. This is necessary in order to build proteins, as DNA is too big to leave the nucleus, so mRNA acts as a physical messenger of the instructions required to build the protein. DNA unwinds and unzips, breaking hydrogen bonds between nucleotide bases. This process is catalyzed by an enzyme. Next, mRNA forms alongside the template strand, using complementary base pairing to copy the code. This process is carried out for one gene at a time, using free nucleotides found in the nucleus of the cell. The nucleotides are secured to adjacent nucleotides through the formation of a sugar phosphate backbone. Once the mRNA is complete, it leaves the nucleus to deliver the instructions to the ribosomes. In translation, mRNA is used to build proteins from amino acids. mRNA leaves the nucleus of the cell and it attaches to the ribosome in the cytoplasm, ready for translation to occur. The ribosome reads the codons on the mRNA, and the tRNA delivers amino acids using their complementary anticodon. The sequence of amino acids is therefore determined by the mRNA. Peptide bonds form between adjacent amino acids, and then tRNA can be returned to the cytoplasm to collect more amino acids. This continues until the entire mRNA code has been translated into amino acids. Note that you do not need to use the term codon and anticodon in your specification. Here is a question like one you might encounter in an exam. The question asks you to describe the process of protein synthesis that occurs in the cytoplasm. This is referring to translation. Some subject-specific terminology which you might like to consider include ribosome, mRNA, amino acid, protein, and complementary base pairing. It is a 5-mark describe question, so you should be prepared to include a fair amount of detail. It is okay to use bullet points to arrange your answer, but you should write in sentences. Firstly, the mRNA arrives at the ribosome. The mRNA acts as a template, and the ribosome moves along the mRNA strand. tRNA brings amino acids to the ribosome. and the tRNA binds with the mRNA using complementary base pairing. An amino acid chain is produced. There are 6 possible options here for 5 mark points, but there are also alternative ways to phrase your answer. The / in the mark scheme shows where alternative answers are possible. Try to become really familiar with the phrasing that suits you best or makes the most sense to you. In summary, protein synthesis is split into 2 stages. In transcription, DNA unwinds and unzips. mRNA forms next to the template strand using complementary base pairing. The mRNA then leaves the nucleus. In translation, mRNA attaches to the ribosomes in the cytoplasm. tRNA molecules use complementary base pairing to deliver the corresponding amino acids to the ribosome, and amino acids bond with adjacent amino acids to form a protein. Don't forget to check out all our other fantastic revision resources here at Save My Exams.
Okay, so we've seen the 2 processes, the transcription and the transcription process and the translation process. Okay, so we say it's taking place in the nucleus, right, and the cytoplasm. Okay, so we say how the DNA makes the mRNA, right? mRNA, and what the mRNA, the DNA unwind the mRNA, right? And how this mRNA binds with or zips with the corresponding strands in the DNA, in the gene code. And then how this mRNA is being moved from the ribosome, because all these ones are taking place in the ribosome, being moved from the ribosome to the cytoplasm where the translation will take place. Okay, so these processes, as we explained, the job of the mRNA, how it moves them, and then before the tRNA that eventually moves them, and then they do base pairing at the end of the day, and then they form protein, unique protein. So at the end of this class, we upload this so that you see the process as it were. Now this is the formation. Proteins are made by ribosome with the sequence of amino acid controlled by the sequence of bases contained in the DNA, within the DNA strand. Okay? Proteins are made in the ribosome, or made by the ribosome with the sequence of amino acids that is controlled by sequence of bases contained in the DNA. Now DNA cannot travel, it's too big in the ordinary sense. It's too large to travel through the nucleus. Okay, so we say DNA cannot travel out of the nucleus. Okay, they cannot leave the nucleus to the ribosome. So what will happen is that it's far too big to pass through the nucleus pore, the small pore of the nucleus, the small holes of the nucleus. DNAs are too big. So what it will do? It will release what we call the mRNA. The small m is the messenger. It's called the messenger. So the DNA itself is too big to leave the nucleus, okay, to pass the pores of the nucleus. So what it will do? It will generate a base code. So the base code of each gene is transcribed into what we call RNA. We've explained that how the RNA is being transcribed, is being formed. So it generates the base code generates what we call the RNA molecules, which we call the mRNA. The RNA molecule is called the mRNA. That's the messenger RNA. Now this messenger RNA is what is going to leave the nucleus. It's what is going to leave the nucleus. The messenger RNA is what is going to leave the nucleus. It's going to fly off the nucleus. Okay? This is the nucleus. The DNA on wing. This is the nucleus. This whole thing is the nucleus. So mRNA is one unit. This one unit. You see, like this U, this G, this U, this A, this C, this C, this A, this U. Each of them is a mRNA. Each of them is a mRNA. Gene is copied into an mRNA. And this mRNA will leave this nucleus through the pore, through the small pore in the nucleus. So it's like a messenger that has been sent a message. Okay? The mRNA will leave the pore, and fly to the ribosome. Okay, it's going to fly to the ribosome where synthesis is going to take place. Okay, now when the mRNA leaves, the mRNA then moves out of the nucleus and attaches to the ribosome. It will leave the nucleus and it will attach to the ribosome. Okay, we already established that the proteins they made by the ribosome, not in the nucleus itself. The proteins are made by the ribosome, right? Or they are made with the ribosome. So, but the DNA itself is too large to leave the nucleus. The DNAs are found in the nucleus. They are too large to leave the nucleus. Hence, they will generate a sequence of base. Since they are too large, they will generate what we call a sequence of base, small unit that can leave the nucleus and move to the mRNA by a process of transcription, and that unit is called mRNA. NA that is called mRNA. That mRNA moves out of the nucleus and attaches to the ribosome. Now the ribosome reads the code on the mRNA in the group of 3. It reads them in group of 3. Okay, the ribosome reads the mRNA that has come to attach to it. It reads them in group of 3. Each triplet of the basis code for a specific form a specific amino acid. Each triplet of bases code for a specific amino acid. So why is the ribosome reading them on basis of 3? Why not singularly? Why is it not reading them one after the other, one by one? Because every 3 forms amino acid. Every 3 sequence that it goes together forms an amino acid. So he reads them in unit of 3. In unit of 3. 3 mRNA that comes to the ribosome will be read as amino acid. In this way, the ribosome translates the sequence of bases into a sequence of amino acid. You see? Once you read them in triplet, in 3, then he's able to translate them to mean amino acid. That makes up a protein. You know, amino acid is the smallest unit of protein. That makes up a protein, but a protein with the smallest unit. Once the amino acid chain is being assembled, like he's continuously doing that. So he have a small unit of amino acid, small unit of amino, like that. So in that sequence, it's going to form a series of amino acid. So it becomes a chain of amino acid, which becomes a protein, which makes up a protein. Once the amino acid chain has been assembled, then it is released from the ribosome so it can fold and form the final structure of the protein. It will be released to form the final structure of the protein. Okay, but it does this by a sequence of pairing, okay, complementary base pairing. Okay, so the ribosome does this happens by doing what we call complementary base pairing, A to U, C to G, A to U, C to G. There's this pairing mechanism that it has a formula, and this is done with the help of tRNA that helps to bring that eventually helps to bring the mRNA to the ribosome. Is that okay? So this complementary base pairing is happening in the ribosome. Okay, so in writing the exam, we must measure complementary base pairing. You must know which unit of the mRNA pairs with another corresponding unit. A goes with U, C goes with G, and A goes with U, like that. Okay, so we must understand this mRNA molecule, how its genetic coding is being carried out. Is that okay? So in this way, the DNA controls cell functions by controlling the production of protein. How does the DNA control the cell function? By just controlling the protein they synthesize, the production of protein. Like I said, protein is almost everything that is happening in our body. So if you lack protein, if you don't have enough protein synthesis, then there's a lot of problem in your system, because the protein may be the enzymes in your body. The proteins not only they are the enzymes in your body, they form the enzymes. The protein form the antibodies. So if you see your antibodies weak, means you're not having enough protein synthesis in your body. They form the enzymes in your body, they form the antibodies in your body, they form the receptor cells in your body, and they form the receptor cells also for your neurotransmitter to happen in the synaptic gaps. in your nervous system. So if you lack protein, you're going to definitely have a lot of issues, a lot of problems. Okay, although all body cells in an organism contain the same gene, all your body cells, they contain the same gene, and many genes in a particular cell are not expressed because the cell only makes the specific protein it needs. The cell of your body only makes the protein it needs. So if it doesn't need a particular protein, that particular cells in the body will not synthesize that unit, that specific protein. Okay, so you know the nucleus that we are talking about and the ribosome, they are in every cell. They are in every cell. So, and there are different cells in our body. Cells are in everywhere in our body. So every cell makes the kind of protein it needs. Every cell code or make the kind of protein it needs. If it doesn't need a particular protein, the cell will not form that protein. If it doesn't need a particular protein, the cell will not make that protein. Is that okay? So that is the brief of this. Let's see if we still have a part to play for this third part. Which protein are synthesized? Which protein? We say everything starts from the nucleus, where the DNA begins to instruct the base code. The base code of the DNA of wind, and the base code generate mRNA that can leave the nucleus, the pulse of the nucleus, to go to ribosome, where there will be complementary base pairing between the base code of one unit to the base code of another unit, which will eventually form and will be carried by tRNA, and which will eventually form 3 pairs of these will form what we call the amino acid, and the amino acid form the unit of protein. Okay, so we've talked about this. Okay, we've talked about this. Now let's take a look at we say expression of a gene means whether that gene is transcribed or translated in a particular cell or not. When you say expression of a gene, it means whether the gene is transcribed or translated in a particular cell. Okay, hello. Okay, so we say expression of a gene means whether that the gene is transcribed or translated in a particular cell or not. Now, most genes are not expressed in a particular cell. Any gene that is not expressed in a particular cell, we can say they are switched off, because that would be a waste of energy, right, and other resources in the cell. Only the gene whose proteins are vital to that cell's functions are expressed. Only the gene whose proteins are vital to the cell's function are expressed. We already explained this. We say the cells makes the particular protein it needs. For what it doesn't need, we call it switched off. It's not expressed. So expression generally of a gene means whether the gene is transcribed or translated. If a gene is transcribed or translated, it means it's expressed. If a gene is not transcribed or translated to form a particular protein, it means it's not expressed. We can say it's switched off. We can say it is switched off. So whenever a particular gene is used to make a particular protein, we say expression has taken place. We call that expression. Okay? Okay, now let's this is the ribosome part of the cell. This is the ribosome part of the cell, and you can see complementary base pairs. How A is paired to U, how C is paired to G, and how A is paired to U also. So every 3 pairing like this will form what in the ribosome, or what we call the mRNA molecule. This is an mRNA molecule. So every time this happens, then we have amino acid. We have a unit of amino acid. We have a unit of amino acid. Okay, this is supported by tyrosine. See, another bond, these are bonds, these are the bonds. Another bond that is holding another 3 peri is called peptide bond. Okay, the peptide bond, you can see the peptide bond is joining the proline and the tyrosine. This tyrosine is holding this unit, and this proline is holding this unit. But the bond between them is called the peptide bond that is holding this unit, 2 units together. Okay, and then here we have the tRNA molecules. The tRNA molecule, these ones, the tRNA molecules, and here also tRNA molecules. Okay, which is carrying this tRNA molecules is carrying this units, small units, okay, of the gene. And here this is a valine. What is holding this particular unit is the valine. This one was proline. This one was tyrosine, and so on and so forth. So each of these are the bonds. Each of these bonds hold these 3 together. Each of these bonds holds these 3 together to form in where it pushes them into, once this is coded, this one takes the next, this one follows, this one join with this, pair with this, and follows. So once this happens, they form, this is amino acid, this is another amino acid that will be formed, and this will be another amino acid that will be formed. So once this happens, it forms what we call the protein, the unit of protein. It forms the unit of protein. Okay, so this is the process how in ribosome how the protein is synthesized using translation. How the protein is eventually synthesized using translation. Now we say the protein may be enzymes, antibody receptors, and neurotransmitters. I think we've expressed this. Okay. Although all body cells in organisms contain same gene, many genes in particular cells are not expressed because the cell only make the specific protein that they need to use. Cell only make protein they need to use. They don't make all proteins and keep them down. They only make the specific protein that they need. Now let's take a look at haploid and diploid. Haploid cells and diploid cells. All humans have 23 pairs of chromosomes, and together there are 46 chromosomes in our body. When we say 23 pairs, meaning one on this side, one on this side, forming a pair. Okay, one on the left, one on the right, forming a pair. There are 23 pairs of chromosomes. Altogether, chromosomes in our body is 46 chromosomes in our body. Most body cells not included, without including the gamete sex cells, we have 2 copies of each chromosome, right? Like we already said. One on one side, the other one on the other side. You have one on the left, one on the right. Okay? Now this will lead to a total of 46 chromosomes. Okay? We already established this. Nuclei with 2 sets of chromosomes are known as diploid nuclei. Nuclei, any nuclei, like the plural of a nucleus. Any nuclei with 2 sets of chromosomes is known as diploid nuclei. Okay? Then the gametes, that's the egg and the sperm cells, only have 1 copy of each chromosome, meaning they have a total of 23 chromosomes in each cell. The gametes, that the egg, the formation, the egg and the sperm formation, which is the new gamete, only have 1 copy of each chromosome, meaning they have total of 23 chromosomes in each of them. Okay? Nuclei with 1 set of unpaired chromosomes are known as haploid. 1 set of unpaired. It's not paired yet. Okay? 1 set of unpaired, which is 23, is known as a haploid. Nuclei with 2 sets of chromosomes are known as diploid. So if you have 2 sets of chromosomes, we call it diploid. Nuclei with 2 sets of chromosomes is known as a diploid. Nuclei with 2 sets of chromosomes is known as a diploid. Nuclei with 2 sets. You have 23 here as one set. You have another 23 here as one set of chromosomes. Let's say you have this and then you have this, for example. Okay, you have this one, maybe here you have another one, you have another one. Okay, like that, 23 on this side, 23 on this side, for example. So nuclei with two sets like this, two sets, they are known as diploid. They are called diploid. They are called diploid. Diploid. Then nuclei with one set, only one set of chromosomes, only 23, which is one set. One set. Nuclei, any nuclei with one set of chromosomes is called haploid. One set is called haploid. Nuclei with one set of chromosomes is called haploid. Nuclei with one set of chromosomes is called haploid. Only one set that has not paired up. One set of chromosomes that has not paired up is called haploid, while the one that has two sets of chromosomes, two sets, is called diploid. The one with two sets is called diploid. The one with one set is called haploid. Okay, so we need to establish this. We need to know this. Okay, nuclei with one set of chromosomes are called haploid, and the one with two sets of chromosomes are called diploid. Okay. Okay, so I think we've come to the end of our today's section, which is part 2 of this class. The next one will be mitosis. We look at mitosis and meiosis in our next class, the cell division. We look at the cell division in our next class, mitosis and meiosis. So with this, we come to the end of today's class. Thank you for joining us.